In this episode of the Paul Simon podcast, I talked to Favour, a bio-ML researcher and final year pharmacy student who is using AI to analyze cancer at the molecular level. His work on AI-based biomarker discovery for meningioma tumor classification achieved over 70% alignment with tumor severity. We talk about how he builds models from genomic data, what this means for the future of medicine, and confront one of the biggest questions in healthcare today: can AI help cure cancer? Could you introduce yourself for those that don't know who you are?
Okay, my name is Favour. Currently, I'm a data scientist. I'm interested in I'm interested in things related to product data science and fun. Will I say fun fact about me outside of work? I'm interested heavily in machine learning and biology. Yeah.
Yeah, okay. So, what are interesting things that you're currently working on or that you've worked on before?
You want context? It depends. It depends.
Okay, in the context of like, um, let's say AI.
Okay. I think at the moment I'm working with my supervisor. There's a project. Sorry, let me put this in. I'm working with my supervisor. So currently, there's a project we are working on. We want to build a framework, a machine learning framework that helps us to see. Um, I don't know. It's going to actually become very technical and very biology driven.
Oh,
It,
yeah,
we
no.
want to see how how different drugs behave in different cellular contexts. And let me explain. We know that um, sorry, I think I have to rewind a little bit.
You're free, you're free.
Okay. We know that in the area of drug discovery, we know that different drugs bind to different cells. A drug will bind
Okay.
to a target and it will elicit an effect. That's why you say, okay, you take a drug and you get well. Understand?
Yes,
This
yes.
exactly. We also know that, and when that occurs, that's like structural binding. The drug will bind to a target, something like that. But currently in the drug discovery landscape, there's no like there's no framework that helps us to see based in different cellular contexts, in different areas, in different cellular contexts. Maybe the cell is diseased, maybe the cell is this, maybe the cell is that. How well will this drug bind to, and what are the structural features of this drug that makes the binding efficient? Do
Okay.
you understand? Yeah.
Yeah, I
So it,
understand.
yeah. So that basically the pipeline I'm currently building, although I'm 70% halfway through the project, we
Okay.
have to we have to first get the data sets. So we have to get cancer cell lines. Different cancer cell lines are in vitro when I say in vitro, yeah, different cells, different cancerous cells, not of the human being, or not inside of, not, how would I say this? Excuse me. Okay, different cancer cells in
Not
a
from
test.
humans.
No, from
Okay.
humans, but you're not testing it on human beings. You're testing it on the cell lines, meaning you're testing them
Okay, okay.
in the test tube.
Like in like a controlled environment, like an
Yes,
isolated test
yes,
tube.
a
Okay,
controlled
understood.
experiment. Understand?
Yeah, yeah.
So, different drugs. We have like more than 24,000, more than actually 40,000 cell lines, and these cell lines, different cancer drugs would have to we would test them on the cell lines to know if this drug can actually inhibit the cancerous cells in that test tube. Understand?
Okay.
So luckily for us, there are open access databases that help us with this data. Then we have to also get some data of two drugs we are looking at, EGFR and BRAF.
Okay,
So
wait, sorry,
EGFR,
so sorry. Can I cut you there before I forget my
yeah,
question? Okay, so what I'm trying to what I could understand from what you just said now is that you're building a framework where you can, should I say, monitor
Not
or
necessarily monitor.
like
We want to,
identify.
yes, something like that.
Okay, identify. So in so basically, should I say the target is like the cancer cells bar, right?
No, 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 no.
Okay.
We know at the moment we know that different drugs have different features, structural features, because each drug each drug comes with different properties. And put it that way.
Okay.
Do you understand? So we want to see what are the specific properties of this drug in
Okay.
each different cell will make them very, very efficient for drug activity, basically. So basically, in let me say in a cell that is cancerous, we can say that okay, uh, this part of this drug, or maybe the hinge point of the drug, or maybe the log P or the lipophilicity of the drug, whatever will make it bind further and increase like drug activity, thereby inhibiting the cancer. So it's more
Okay.
of we are trying to model um, how drugs will act, assuming it was in the human body using machine learning.
Okay. 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 Hmm. Okay, I get that. I get that. So, okay, so it doesn't necessarily have to be only cancerous cells, but you can also
Yes.
test with like, oh, okay,
Yeah,
okay.
that's why
So,
it's a framework.
okay, so currently you're just working with cancerous cells now, right? Okay, okay,
Yes.
okay. So you can continue from where you stopped.
Then after that, that's basically much of it. That's actually what I'm working on currently in the area of research.
Okay, okay. So what exactly is like a finished what
Project
do you
to
call
me.
it now? Yeah, exactly. To you in that regard now.
Um, a finished project to me is did we achieve our hypothesis? So basically,
Which
my project,
is
which is, um, are there some features, are there some structural features that we know that is kind of new that we could like actually like add to literature?
Oh, okay, okay. So you're basically like looking for like gaps in the current literature
Yes,
that you
yes,
guys have
yes,
now.
yes. And
Oh, okay,
we are
okay.
using the project to test that. Once we are done with our analysis and the results of of our models, we can now say or infer that oh yes, our hypothesis was correct or our hypothesis was wrong, and we'll still write a paper about that. So it's basically
Okay.
you start the project, you complete it. The way you know you have completed it is if you have accepted if the result of your analysis, if it like agreed with your hypothesis at the beginning or
Okay, okay.
rejected it.
Yeah.
So once you're done, but it's very important. Honestly, it's very important. Every research has to have a paper to that. So
Okay. So my question, I have lots of questions, but I'll try to limit them. So, why aren't you also testing out like other types of diseased cells? Like, why is it only cancerous cells that you're working on?
because it's easier to get data sets or data sets related to cancer
Okay,
than other
okay.
diseases.
Oh, okay. So they're more accessible than
Yeah,
other
they're
types
more,
of data sets.
yes, they're actually more accessible.
Okay. So you mentioned models. What exactly do you mean by models?
I think this is actually this is a very tricky question because I
Okay, wait,
can
wait. Okay, you
describe
know
it,
what?
but I cannot explain it.
Just
I
correct
think
me. Okay. I think let's try to get there. So, okay, you you do the experiment, you extract the data, right?
mhm.
So what do you do with the data? How do you go about it? How do you use the data rather?
It depends. It depends on your question, the research question,
Okay.
the question, the class question. On mine, for mine, the major question is, does the structural features of each drug change in different um, cellular context? Meaning,
Okay.
does each property of the drug based in different cells, do they change, do they like affect drug binding, which will lead to drug activity? Understand? So
Okay.
from that, when we got three important data sets for this from three different databases. We had to do data integration. Understand? Because since they come from three different places, we have to find a way of adding and making everything integrated and a one uniform mega data set. Understand? Looking for like um, a column that will join everything.
Okay.
Then after
Okay.
that,
Sorry. So, how does a data set look like?
rows and columns with
So,
rows
like,
and
what
columns.
exactly? What are the values exactly? Like, what? Like, what is the value? Like, value. Like, okay. Let's say. Do you understand my question?
No, 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 no.
Okay, so let's say in a cell now, in a table cell, what are you? What is it? Numbers? Is it letters? Like, I want to really go into that.
We know how like a spreadsheet is like. A
Yes,
spreadsheet
yes.
has tables, columns, um, anything. I
Can
would
be
say,
anything.
yeah, it can be anything, but it's always arranged in rows and columns. So
But
they,
that's like I'm asking like what is the data type exactly? Like is it like okay, let's say is decimal numbers, like what do those decimal numbers mean? That type of thing.
that's why I say it depends. It depends. It depends on where you're getting it from.
Okay.
For us now, we got data set on different drugs tested in different anti-cancer drugs tested in different cellular cell lines. So basically, what we might be seeing there is the drug name. Understand? A column should be
Okay.
the drug name.
Yes.
Understand? On that column should be the IC50. IC50 is the drug activity, 50% of the drug that can actually like inhibit something.
Okay,
But there are
okay.
other columns that you see, but it all depends on
What you're
what,
trying to extract.
yes, what you're trying to extract or what you're trying to achieve,
Okay,
or
okay,
the
okay,
purpose
okay.
of the data.
Okay, okay, okay. That's clear now. So okay, exactly. I want to sidetrack a bit now. What's the difference between um or is there even a difference between a data scientist and a data engineer?
There's a huge difference. Um, a data scientist majorly like builds models. His work is majorly about like um, I call it data storytelling from different angles. Understand? It's someone who tells you, helps you to understand. Um, wait, wait, I need to gather my thoughts. It's someone who
Take your
helps you to
time, take your
understand.
time.
Yeah,
Yeah.
I'm looking for the right word. I'm looking for how to word it correctly. But majorly somebody who helps you to get understanding of what your data is saying. I'll put
Okay,
it that way.
okay.
While a data engineer is basically, my mind is a software engineer who instead of like writing code for software, writes code for data pipelines.
Okay. Then what's now the difference between a data engineer and then ML engineer? Or is there even a difference?
There's a difference. There's a difference.
Okay.
Um, data engineers necessarily building the infrastructure that moves data from one point to another. That's just a very very like very basic way of saying it. While ML engineers build models, they build these things or these algorithms that help us to predict, answer questions, and they deploy that too.
Okay, okay, okay. Okay, now going back to this current project you're working on now. Um okay, so you get, okay, I was asking you about what exactly do you mean by models.
Mhm.
So you get your data, you analyze it, you do whatever you want to do with it. So um okay, exactly. Okay, let me go back a bit. So when you get your data, what do you do from there?
You clean. I think
Okay.
you're going to clean because um, you clean. First of all,
Then
you're
after
going to
cleaning.
get after data cleaning. You do what we call feature engineering. So basically, in feature engineering, you are necessarily like choosing or selecting or filtering down the necessary things you need to feed your model. So um, excuse me, sorry. In ML lingo, there are different types of model or different types of way models learn. There are supervised learning models, unsupervised learning models, semi-supervised learning models. Understand? I don't want to actually go into detail about that, but
No,
I'm
don't,
getting
don't. No need, no need.
like getting too technical. But definitely, but what the first step is actually like exploring what your data looks like. So if you get a data set, you're going to like want to know, you want to see the columns and rows, like the first five rows, and what each column is saying, so you can have an understanding of what it is. Then you can also like do like some median statistics to see if there are like null records, if there's any record that is like that has like blank spaces. Do you understand?
Okay.
Then after that, you now do feature engineering. Feature engineering is mostly if you want to build a model, you have to like engineer your features. And when I mean engineer features, is engineer the columns you need for your prediction or your classification. Do you understand? So then, okay, let me say I want to predict the price of a house. I want
Okay.
to predict the price of a house from a data set that has different columns like area of the house, number of people living in the house, number of rooms, different things. Understand?
Yeah.
What is the most important thing? What is the most important thing that will help us to um, necessarily predict the price of a house if you're showing it to a model? And a model is like an algorithm, a mathematical algorithm that helps us to predict things. Obviously, it's just basic equations and vectors. So if I'm to ask you, what is um, what is something that what is some excuse me, how would you know the best performing performing person in your class? What would they
Great.
want? The grades,
Great.
exactly. um so yes, so for to predict the house, to predict the price of a house, it could be the number of rooms. That could help us to know, because obviously if the higher the number of rooms, the higher the price, basically, I think. It could also be the square meter, the area of the
Yeah, yeah.
house. It could be so many things. So in feature engineering, you're necessarily like trying to pick the necessary things you need for your model to help you to achieve your target.
Understand, understand. So after feature engineering, what happens?
After feature engineering, based on how well your model performs, the regression coefficient, there are some parameters we use to check if a model is performing well. That is the R squared error, the mean squared error,
Okay.
um, the precision recall. Um, Once again, so many things, but most people focus on the R squared coefficient, which is if
Okay.
it's closer to one, it means that your model is
Okay.
actually like accurate. Understand?
Okay. Okay.
So
Okay. Okay.
0.9 is much more accurate than
Oh,
0.8 or
okay. So in
0.7.
a scenario where you get like 0.1 or 0.2, what do you do?
Mm.
Do you do everything again or
No, no, no. You tune your features, you tune your parameters, you arrange your weights.
okay, yeah, 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 yeah.
You just
Okay.
you go back and arrange the model. The You just fine tune your model
Fine
basically.
tune, yeah.
Yes.
Okay, with the already existing. Okay, that makes sense.
Mm.
That makes sense. Okay. Is there a scenario where this will I call them okay, you said you test it with these parameters now. Is there a scenario where you'll be like, oh wait, this is not a fine-tuning problem. I have to like literally get new data to redo this whole thing. Is
Mm-hmm,
there ever a
mm-hmm.
scenario like that?
Yes. When either when the model is overfitting. So here
Okay.
in this scenario, overfitting is where a model, because two things. If you want to predict something, if you walk into a room, you see, if I want to ask you to, okay, based on the number of people here, can you predict who is the top scorer? Understand?
Okay.
And I'll give you like a kind of like a hint. I'll show you
Yeah.
the maybe the top people in the class. For example, I'll give you a hint. Then after that, I'll be like, okay, using this hint, let's walk into on that class based on what you have learned, based on the patterns you have learned from this hint. Can you, in this new class, can you help me to predict who are the top performing students, or who is the top performing person in this class? Do you understand? So using that analogy, overfitting is a scenario where a model learns the patterns
Okay.
very well, and doesn't do well in the new data set, which is the training, sorry, the test data set. So in the training set, the training
Yeah.
set is the one you give the other one, the other you divide your, sorry, I'm talking too fast. Uh, so basically, you have
It's
your
fine.
data set.
Yeah.
You either divide a 70-30 ratio. 70, you train your model on the 70 and ask it to predict on the 30, or either 80-20. Majorly, the training set is 80% of the data set. Sorry, training set is 80% of the data set, and the test set is 20% of the data set.
Okay.
Do you understand? So your model is being trained on that 70 or 80%. Then the model is now asked to now, based on the targets, can you predict on this separate 80% that you have not seen, you know nothing about, based on the patterns you have learned from the training, please predict
Yeah.
on this new one.
Okay. Yes, I understand. Okay, that makes sense. That
Mm.
makes sense. Okay. So after you you're getting like good results from this parameter you're testing your model with, what do you
Mhm.
do after that?
I don't understand. Come again.
Okay, I was asking like um after you test this model here with these parameters, like you said, the R square and all those other things, what do you do after that?
You 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 check after the results, after you've gotten your results, does it align what you want? Understand? It's
Okay.
based on, does it actually align what you want? If
Yeah.
it aligns, you can go further. You can now deploy your model if it's actually a model you want. But most times, most people really don't necessarily get what they want from the first try. So they go back, they retune, they change some parameters, they put some parameters
Mm.
forward. They just do some basic mathematical stuff in code actually. Understand? So here and there, they tweak it. It's just necessarily like tweaking
Okay.
the parameters of the model to actually
To
then
get what you want.
get what you want, basically.
Okay. Okay. So I still have so many like training questions, sure, but let's talk about like hardware now. How how do you navigate hardware? I know fine tuning or doing all these things is should I say it's expensive in regards to compute. So
Yes,
do you
compute
like
resources.
is it like a bottleneck for you personally, or you just try to um not do too much? Like how do you navigate hardware?
Okay, if I understood your question, this applies to like GPU.
Yes, exactly,
So
exactly. Yeah.
yes, so there are different type of models require different type of compute resource.
Yes.
That was the most. So basically, like for deep learning models, they're not necessarily like traditional machine learning models. They are they are much more. Um, I'll use the word abstract. I'll use the
Okay.
word abstract. I'll use the word abstract. So deep learning models require much more higher computing power than traditional machine learning models you can run on a CPU. So I think for example, for my project,
Yes.
um, I was analyzing over 70 videos um, of rats. I'll put it like that. So my project required, we wanted to see if the extract of our plants had like a pharmacological effect. So before now, in traditional pharmacology, what we do is you give the rat the extract, then you watch the behavior of the rat. Do you understand? But currently there's a rise. There has been there has been a rise in computational pharmacology, new methods or AI in computational pharmacology, where you can use post estimation tools or AI methods to track the movement or behavior of a rat in video. Let's say, understand? So I did that. I got my experiments. I got my videos. Understand? And
How many videos?
75 videos.
Okay,
Understand?
75. Okay.
Up
That's not too
to
much, then.
that is not too much, but these videos like were like 300 MB, 500 MB.
Oh, okay, okay. They were not
Total,
like short videos. They were
yeah,
long.
yeah.
Okay.
So in total, I had up to like 25 MB, so 25 gigabytes worth of videos. Yeah, I had to like train deep learning models on. So I think last year I started working on this around December, and this video, I was using Google Colab. So Google Colab gives you free GPU hours of like say 1 to 2 hours. But my videos were a lot that like within 30 minutes I would use it up, and I created like 3 Gmail accounts. Like but it was not like
Yeah, yeah, yeah.
it was not right. I would just say it was not right. So I used Cargo. So I don't know if you know Cargo. Cargo is what's Cargo again? Like I know what Cargo is, but what's Cargo? Cargo is a
I
platform.
beg. Okay, let me
Yes.
check. Cargo. Can you spell it?
Kaggle.
Kagg. Oh,
L.
cargo. Okay, okay.
Yes.
Oh,
So
yeah.
Cargo. I used Cargo. Cargo. Cargo gives out 30 hours per week of free GPU hours. Understand? 30 hours uninterrupted. Or is it 30?
Oh,
Yes, it's 30 hours. So I use Cargo to do that.
cargo. Cargo, right? This one.
Yes, yes, yes.
Oh, really? You said how many GPU hours?
30 hours.
30 hours. Wow. I like is it per month
No, it's
or
per week. Per week. So if you're exhausted, you don't get to use GPU, you don't get to use the GPU resource. You now have to use CPU. And deep learning building a deep learning model or fine-tuning a deep learning model on a CPU is as slow as a slot. So
yeah.
GPUs are faster. Understand? GPUs are very faster.
Oh, this is crazy. Okay, you said 30 hours is really generous, so
Mhm.
it's really generous. So okay, so you use Kaggle then um okay, you use Kaggle to do the um to handle the seven is it you said 25 watt of
Yes, I use
25
Cargo to
gigabyte of videos. Okay, okay.
yes, I use Cargo to make it easier for me to complete and analyze the videos on time because my supervisor was already on my neck. to get my results.
Oh,
So, but I'd use CPU. I was using CPU, and one video would have taken me one day. One day. I use the CPU on my laptop. But with Cargo, I
wow.
completed everything in just 3 days, the whole 75 videos.
Wow, that's decent. Oh, that's really decent. Okay, so um I'm trying to remember what I asked you. Okay, yeah, I was asking about how you navigated how the okay, that's actually a perfect question. That's a perfect answer because normally the normal excuse that people have when they want to really break into ML and deep learning is that it's expensive. You get
Yes,
compute and all that.
expensive.
So
Yes, very
exactly.
expensive.
So this, trust me, this your should I say insights. Me personally, like I wanted to use one on AWS, but AWS can be very complex and
Yes.
confusing. So
With the,
Kaggle,
I think AWS
yeah.
has something like AWS instance E2. I don't know if it's E2 instance or
EC2,
something. Yes,
EC2.
EC2, yes, EC2.
Exactly. I use EC2 and um they'll they'll say it's free, yeah, to some extent. Then you now find out that at the end of the month you get like an email billing you $29 for. So that's exactly what happened. You get so Kaggle is like a perfect example of how people can actually start and really break into ML and AI. That's bro, this is helpful, trust me. Okay, so okay, exactly. I told you off camera that I wanted to really talk about a should is it was it a research or a publication about um
Okay.
cancer? So just give us like an overview, then we can just start dissecting from there. Because personally, I think it's very, very interesting. So yeah.
It depends. It depends. I should talk about cancer or I should talk about what in
Okay, you know
general.
what? Okay, you know what? Okay, to prep us for you going in depth. No, not you going in depth, but to prep us for like the you explaining the research paper. Yeah,
Mhm.
just give us like a overview of um um cancer. Then you cannot transition into okay what you're trying to do in regards to cancer. Do you understand?
Okay, I have to pull up. Let me pull up my. Let me pull up to the paper. I think you're referring to.
Oh, perfect. Okay, can you see it?
Yes, I can see it. Wow.
Comparative analysis of
And
tumor mutational
mutational.
burden and copy number alterations in. That's a lot of
That's a lot of
that's
words.
heavy. Is a mouthful. So
Yeah.
you can just give us like a um
An overview of what I did.
overview. Yes,
Okay.
overview. Yes.
So here I did an internship at an online virtual internship at Genomic. So Genomic is like a bioinformatics virtual lab. They
Yeah.
have different cohorts for different things you want to learn about. So for this, I worked on studying meningiomas. So meningiomas are the most common type of brain tumor. So here I
Hmm.
wanted to understand how aggressive, what makes some of these tumors very, very aggressive than others. So the key idea here was that instead of just looking at the tumor under the microscope, like
Yeah.
normally brain tumors, most brain tumors or most brain cancers, the way they study them or they look at the aggressiveness of is studying them under a microscope. You take some things about the tumor and you just use a slide, just normal microscopic stuff. So here,
Yeah.
instead of looking at that, instead of we analyze the DNA because what's cancer basically? Um, Cancer is in the most plain word, an abnormal cell. Do you
Mm.
understand? Cancer is not necessarily an infection or not necessarily like any other thing. It's just a cell that just became wayward. I'll put it that way. Understand?
Okay.
So instead of looking at the tumor under with this knowledge, instead of looking at the tumor under the microscope, we analyze their DNA because every cell in our body has its own DNA that's been produced by the chromosome. Understand? So we looked at two major features. First, we looked at the tumor mutational burden, which is basically how many genetic changes that tumor has. So the more changes, the higher this number, the 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 possibility for that cell, for that tumor to be very very aggressive. Understand? So
Wait,
if you
sorry,
have
sorry. Can you say that again? The higher what?
the higher the total mutational body number, the more
Okay.
aggressive that tumor is
Okay,
going to be.
can you okay, is that like the degree of aggression?
Yes, degree of aggression. That helps you to tell
Okay.
you, how much, what stage that cancer is, because most cancers that have like a high TMB number of, let's say, what percentage, a very just high TMB number and a B3 number would definitely be grade 4 or grade 6. Grade 4 cancer. I know that grade 4 cancer is more aggressive than a grade 1 cancer. Understand?
Okay.
So
Okay.
this TMB or tumor mutational burden number, it just tells you how many genetic changes that tumor has. If a tumor has so many genetic changes, that tumor is very aggressive. That's one. The second criteria is the copy number alterations, the CNA. So it tells you the sections of the DNA that have been duplicated or missing in the tumor cells. Like I told you that cancer cells are very, very stubborn. They are wayward children, meaning that instead of them having like, let me say, A B C to Z, like A B, the 26 letter alphabet, and every,
Sequential.
yes, sequential, they might have A B C to Y and delete Z,
Hmm.
or they might have ABC to Z, but they might not be OMA or something like that. Just know that cancerous cells, there's always something going on. There's always an alteration, either a deletion or an addition. Something
Okay.
just is
Yeah.
going on. Understand? So this DNA helps us to tell you the sections of the DNA that these alterations are already like going on. So I think of it like looking for spelling mistakes or missing pages in a book. The more mistakes or missing pages, the more aggressive that tumor is going to be like. Understand? So if you get a book, you're looking for, are there some spelling mistakes? Are there some missing pages? So if there are so many of this, it tells you that this tumor is very aggressive and it's going to kill people faster. The same.
Okay,
So
sorry. When you mean aggression, do you mean the faster it spreads?
yes, the faster it spreads, the faster
Okay, you
it
mean
becomes
the,
very, very deadly.
okay, okay. So basically, what makes cancer fatal is the rate at which it
It's
spreads,
yes, the
it
rate at which it
replicates,
spread.
something like.
Yes, duplicate, yes.
Okay, okay. So, okay, okay. No, you can go on.
So after that, so how did we go about it? We looked at 123 tumor samples gotten from the research conducted by one agency, sorry, one lab, sorry,
What type of tumor?
meningiomas,
Exactly.
mening,
What's for those that don't know what that
okay,
is, including me,
meningio,
what is that?
okay,
Okay.
meningiomas are tumors of the meninges. You guys know what like meningitis? What's meningitis? So there's currently like in the human body, there's a layer, covering our spinal cord. Like this is getting so much so or it's getting physiological, so anatomy like
Is it a tissue?
yes, a tissue. Yes, thank you. I was looking for the word tissue. So it's a tissue
Mm.
that covers um our spinal cord. Understand? So um meningiomas are the tumors of these tissues. Understand? I know that the spinal cord is connected to the brain. So That's why it's one of the most common brain tumors ever at the
Okay,
moment. I
so
think
basically a meningom, can you pronounce that again?
meningioma,
Okay, a meningioma is basically a tumor that grows um on
spinal
your
cord, yeah, on the
okay,
tissue
okay.
on the tissue covering your spinal cord.
Okay. So what was the question again?
So I
Exactly.
was
You're saying that you collected this tumor cells.
yes, so we got like
Okay.
123 tumor samples from the University of Toronto. I think the Department of Biochemistry. Then we found out that what we found was that tumors, um, after doing our analysis, we found out that tumors that were more aggressive had twice many mutations and more DNA changes than the less aggressive ones. Like, let
Sorry,
me say,
can you say that again?
tumors that were more aggressive were twice
Mm-hmm.
more, were two times more, had two times many more mutations, meaning that
Okay.
a grade four, a grade one, a grade three will have two times more mutations than a grade one. Do you understand?
Okay.
And also the same thing as the DNA changes, like I told you earlier. So, and we also did something very, very important using this information or using these features. We trained a computer model to predict, to now tell us, okay, we have done our analysis. Can you now help us to predict if we are still right too at the same time? And we also trained the computer model to predict the tumor severity. And what we found was that 76% of the time, um, a tumor is also like aligns with our initial findings. So basically, machine learning is also agreeing with us too. I don't know
Okay.
if that makes sense.
I mean, okay, wait. Basically, the model. What was the model doing exactly?
The model wanted to predict the TMB number, the aggressiveness of the tumor
Of the
using
tumor
these
cell.
features of the tumor cells, using these critical features like TMB and CNE. Understand? So yes,
Oh, okay. So, okay, what were the okay, when you meant the models were agreeing with were giving you expected results, yeah. What do you mean by that exactly?
so we wanted it to confirm our hypothesis. And what's our
Okay.
hypothesis here? Does increase in TMB lead to tumor aggressiveness? Understand? That was just it.
Okay.
Do you understand? We know
Yes,
that
yes.
scientifically, increase in TMB should increase should correlate with tumor aggressiveness. So we just wanted to like confirm
Yes.
that.
Confirm it with your models.
Yes, with the model.
Oh, that makes sense. That makes sense. Okay, so basically it was a it was just basically something you wanted to prove. No, it was basically using a model to prove what already existed out
Yeah.
there. Would you say that?
Yes.
Okay. So exactly. Okay, this is the perfect segue to what I want to discuss with you now. I don't know whether you've seen like debates debates out there saying um AI is going to revolutionize healthcare and medicine. So I think I mentioned this off air. So what does it mean to cure cancer? What does it mean to cure cancer? Because I think that's a very blurry. It's a very blurry topic. You understand? What does it mean to cure cancer? Let me react to the question. So there is this. I won't say debate, but there are questions around um questions around how AI is going to accelerate new discoveries in healthcare, basically. So Before I ask you the question, I want to ask you now. Um, I want to know, what does it mean? Okay, when you what does it mean to say that um God, I've forgotten the question what I want to say. Okay, I've not forgotten it, Sha. I'm just trying to ask you the best way. Okay, is there such thing? Okay, what does it mean to cure cancer exactly? What does it mean to cure cancer?
So many things.
Okay.
I would say so many things, but disclaimer here, I don't think AI can really like cure cancer because I don't think people are wording it correctly. That's the but AI can aid in helping us let's say can aid us in helping us find better ways of controlling it or
Oh, hello, Favour.
can you hear me?
Can you, yeah, can you like go back like 10 seconds? Like your voice
Okay,
was cracking.
okay. I said uh, a major disclaimer here. AI will not cure cancer.
Okay.
It's I don't think people are getting it correctly. It's so many things go into the supposedly cure for something, for a disease. So many things, clinical trials, testing, lab experiments, so many things. But what I would say is that AI will help us to or be like a strong assistant for us. It make it faster, make it more efficient in getting to that stage.
The stage being a cure to cancer.
Depends on what cancer.
Okay. So exactly. So are you saying that there are cancers that can be cured? And in my definition of
Yes,
cure, I mean completely eradicate it from you.
yes.
Okay. So can you give us an example um of what does that look like?
So let's say, okay, so the ones that are not very invasive or very aggressive.
Okay.
Example of this type of cancer is I think this is prostate cancer.
Okay.
It's not so aggressive compared to other ones like let me say breast cancer or even brain cancer. There are different types of brain cancer that are very aggressive. I think the one that is actually very aggressive is like colon, the pancreatic cancer. Those ones that happen in the systems that um, you cannot even use like X-ray scans or imaging scans to detect, that you don't even notice that you actually have this until you're at like the final stage of your life or final stage of the cancer progression. Do you understand?
Okay, that makes sense. Yeah, that makes sense. That makes total sense. So basically what you're saying is that AI, okay, you said AI can't really necessarily give you the cure to cancer. Are you saying that AI can't, do you think AI can make new discoveries in healthcare and medicine?
Yes, exactly. You can actually make new discoveries.
You mean on its own, or
No,
AI help,
you you have to feed something into the machine because at the end of the day, those are computers, garbage in,
like
garbage
training
out.
data?
Yes, training
Like
data,
this, okay. Okay, so basically
training it,
what you say, okay, training it, yeah. So you
training
cannot be
it.
like,
After training it,
okay, sorry. So like I'm okay, let me like dim it down to like the simplest case scenario now. You cannot go to like like in the coming like next 10 years now, let's say 100 years. So is there a possibility where I'll be like on ChatGPT now? Oh, what is the cure to this? Like let's say HIV now, and like there's no general consensus of a cure. Do you think ChatGPT will be able to generate a new discovery, generate an actual cure
no,
to
no,
that
you cannot.
disease?
No, you
Okay,
cannot.
I see that it can never. So
I
basically,
would say at the moment,
what? Okay, at the
at the
moment, no.
moment, no,
So do you
because
think we'll ever get to that stage?
I think we'll get to that stage. It depends on what stage.
Okay, stage generating new discoveries
Yes, obviously,
that
obviously.
actually yield results.
Um, currently there has been a very uptick in generative AI, where AI helps you to maybe generate drug molecules that are actually like faster, or
Oh, wow.
what's even going right now is digital twins. AI can model a human body,
Oh,
it
wow.
can actually model a human body or the actual cell that you're trying to target or you're trying to treat. These things are things that in the past would have to go to the lab and use animals and run too many tests. It will actually take longer time. But with the use of generative AI, these things are now like compressed to make it faster. Um, Whichever thing that AI gives you, you would have to still test it in the lab, conduct some tests. Um, um, it takes time. I think it takes time,
Takes time.
but the creative things or the things that require us to do the long arduous task before now will now be compressed to make it faster and efficient.
Oh, okay, okay, okay. So, okay, you said that there's a point where AI will be able to reach a point where um you don't even need to like run tests on its theories or discoveries. It just gives you. Okay, what I'm trying to say is that there's a point where, okay, do you believe that there's a point where you literally just have to ask it a question, like there's no training data on it at all, there's no general consensus on, okay, there's a cure for this disease. Okay, what I was asking is that, yes, obviously, Gen AI, they'll give you like advantages, or no, it will replace repetitive workflows that will require maybe a doctor or a scientist to, let's say, execute a hypothesis here. But is there a scenario where the AI will actually do all of those? Like you do the testing and you give you a new discovery that was never discovered, that was never even that has never been thought of. Do you get my question? And if okay, and if there's a point AI will reach that stage, how far off? Like how many years do you think? Okay, and can you hear me? Yeah, and I'll ask it, if there's a point in which AI can reach that stage, how far off do you think we are from it? Like, how many years do you think it will take? Like, is there a timeline to which scientists will not have to do their tests and all those things? They can just literally go to, like, let's say, ChatGPT and just ask them, and ChatGPT do everything for them, give them a new discovery. Do you get?
Yes, okay, but I don't know. I don't think I actually have an answer to this question,
Okay,
because
so okay.
the way my mind is the way my mind is explaining it to me. I don't know if that's what you're saying, is that um we won't be needing human beings that will, in the process of this generation.
Yeah, please can you say that again? Like you got cut off.
So I said that I don't think I necessarily cannot answer that question, in the sense that I, at the moment, I'm not so sure, because I don't think I understand what you're saying. My mind is telling me that you mean there'll be a time where um, AI will handle everything from the testing to the deployment to you name it, which I don't think is possible.
Okay. So you're basically saying that you'll never be possible, right?
Mm,
You're not sure. Oh,
I'm not sure.
you're not sure.
That's
Okay,
answer.
okay. Okay. So it's only 1 hour. let's round up.
Mm-hmm.
Um, from what I can remember, you've been in this journey for like let's say data science in general and machine learning for like it should be like 2 years now. Is it 3 years or 2 years?
No, 3 years.
3 years. Okay. So if there was one advice you would tell Favor from day one, like okay, I wanted to embark on this journey. If there was one advice you would tell him, it has to be only one. You can't say multiple. You can't say too long. You have like let's say 1 minute to talk to him. What would you say?
Um, can you hear me?
Yeah, I can hear you. What will you tell him?
It gets better. That's what I would say. It
I
gets
guess it.
better. Yeah.
That makes sense.
Trust me, it gets
Okay,
better.
it gets better. Hmm. Okay, I think that's all. That's all we have left. Favor is okay. Exactly. Do you want to say something?
No, 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 no. I'm just going to say like I want to say thank you. Um, what else again? Nothing, nothing in general.
I wish I really could go on, but I don't want to take your time. I know like you have
Yeah.
things you're doing, so